Hello everyone. I was given a topic of a comparison of COVID-19 vaccines when I was attending refresher course in biological sciences, which was online because of the COVID-19 pandemic. And I prepared for the seminar topic and I thought of putting that on my channel so that even others would get benefited. So let us start with the concept. And I'm going to elaborately talk about this concept in 4 points. First, we will briefly brush up with the basics of what is this virus all about, what is its origin, how does it look like, what is the structure, its genetic makeup, the size, shape, and all other important information. In the second part, I'm going to talk about the life cycle of the virus, which both these points are going to be really very quick and we are not going to go into deep aspects of it because the main point comes now in the third part, which is the vaccine development strategies. Once we discuss about the conventional and modern vaccines used against SARS-CoV-2, we will then conclude our discussion with comparing these vaccines and the safety issues and other challenges in vaccinations. Uh, Before I proceed with the first part, let me tell you, you would require to know the basics of virology as well as a little bit of immunology. So, like what is antigen, what is antibody, what is a virus, and how does it look like. So, for basics of virology, I have another video on the same channel. You can refer to that Virology Introduction to Viruses. Let us continue with our discussion. We all know that this is not the first time that coronavirus is infecting us. In the history, in 2003 February, we have had a severe acute respiratory syndrome virus, which was the first one of its type. And in 2012, we had another of its type, which was called Middle East Respiratory Syndrome virus. But yet, I would not say that this is the third coronavirus that we know. It is actually the seventh coronavirus that we know. As you can look into the diagram, there are certain low pathogenic coronaviruses which were identified earlier, and you can trace them back maybe up to 1962. Its size ranges about 80 to 120 nanometer, the diameter, and it has got a genetic material which is positive sense single-stranded RNA. Now, what do I mean by positive sense? It is the RNA that, when it enters inside the host cell, can be utilized directly as mRNA for the production of viral proteins. In case of viruses which have negative sense RNA, there involves one more step of converting that negative sense RNA into positive sense RNA, and only then a virus can synthesize its protein. Okay, and a total genomic size is approximately 30 kilobase pairs. We all know this, that most of the people do not have a very intense illness. It is ranging between mild to moderate, but yes, if the person is old enough to not have much immunity, or if the person is having some underlying medical problem, then there might be serious consequences. With this background knowledge, now let us take a closer look at the proteins that are present on the viruses, which are called as structural proteins. You can correlate with the color of the proteins. So the yellow colored proteins, or which we know it as spikes, and this is why we call the virus with the name corona. Corona reflects with a crown-like appearance, So that particular protein, we call it as a spike protein. And the blue proteins, I hope you can see that. The blue ones here are known as membrane proteins. And then we have red ones, nucleocapsid proteins. And then we also have envelope proteins. Now, if I ask you which, according to you, would be more in number, the obvious answer you would tell me is spike protein, because that is how we draw a diagram and that is how it looks like. But that's not the case. You'll be surprised to know that maximum number of proteins are membrane proteins present on the viruses. Below that, you have spike proteins, and envelope proteins are the least ones. Okay, but among all these proteins, we are going to target more on the spike protein. We're going to talk more about it. And why we need to talk about spike protein is because that is the one which plays a very important role in the first step of virus life cycle, which is called as adherence. Okay. Now, any virus, be that an animal virus, be that a plant virus, or an bacterial virus, bacteriophage, which we call it as, that follows a cycle, right, inside the host. So it will have to adhere to the cell. It will go inside the cell, which we call it as an entry or penetration, and then it will open up and it will release its genetic material and other enzymes, and then it will take charge of the host cell, replicating its genome, producing its proteins, and then those proteins and genome get assembled together, and then the viral particles gets released, which we call it as a progeny viruses, because that's a next generation of viruses, isn't it? So this particular life cycle starts with the basic step of adherence of viruses to the host cell, and that is where we are trying to block the infection. We don't want viruses to actually adhere to our cells. So how do we achieve that? Is using the spike protein. Let us first understand what is this spike protein. Okay, so we have got two domains on the spike protein. As you can see, there is S2 domain and there is S1 domain. So if you can see this part here, which is like a stick, okay, so that part is called as S2 domain, which is kind of a transmembrane, okay, whereas the upper one, which looks like a crown on the head, isn't it? So that particular part is called as S1 domain, which has got something called as receptor binding domain. So what is the receptor? It is the portion which will be present on the host cell, host meaning human here in this case, and these receptors are going to bind to this, and because of this, adherence is going to take place. So what is the receptor? Receptor is ACE2, ACE2 receptor. Now what is this receptor? This is the receptor used in renin-angiotensin pathway. If you have ever noted about it, you know that this pathway is responsible for keeping control on the blood pressure, and a very important pathway wherein it plays a very crucial role, and many organs are involved in maintaining the blood pressure throughout the body. There is kidney, there is lungs, there is liver, and there are lots of communication. Even adrenal glands are involved. So ACE2 receptors are present on these organs. As you can see, this blue colored, purple colored thing shown here is nothing but the receptor, where it is going to bind with this particular um, viral spike protein. So this receptor is present on the epithelial cell lining of the lung, heart, kidneys, intestines, and so we have plenty of ACE2 receptors to which our virus can bind. Okay, with this, now understand the life cycle of the virus. You can see the virus. Let's consider this line that represents the cell membrane of our host cell, and on the cell membrane, as I just told you, there would be a receptor present. This receptor is called as ACE2 receptor. Okay, and ACE2 receptor is going to bind to that S spike protein, and that virus is going to push it inside, forming a vesicle. Okay, it is called as endosome. So this endosomal vesicle is going to have the viral particle, and then there will be fusion of endosomal membrane with the viral membrane, as you can see in this diagram, and then the genetic material will be released. Now I told you that the genome is positive sense ssRNA, which directly acts as an mRNA for the viral protein formation, that is translation. Okay, so no need to go inside the nucleus for this virus, and thus making the life cycle very, very simple. Now from here it follows two ways. The first proteins that are generated are generally for the replication of the genome as well. Okay, so the enzyme that importantly helps the viral genomic replication is called as RDRP, because it's human host cells. We have generally DNA dependent RNA polymerase. I hope you know the central dogma of life. It gets its own enzyme, and that enzyme is called as RDRP, which is nothing but RNA dependent RNA polymerase. Okay, and with this, it creates a negative sense RNA. With that, again, it creates a positive sense RNA for packing it into progeny particles, progeny viral particles. And here is how intracellular trafficking takes place, where all the viral proteins are regenerated, and assembly of a genomic material that was just replicated by the enzyme, and all the particles is taking place here with the same endosomal vesicle formation, and that is going to fuse with the membrane and release the viruses outside the cell. So this is the one cell life cycle that I've shown you. Like this, at a time, many cells would be damaged, and they'll be releasing more and more viral progenies at a given time. Understanding the viral life cycle, we have finished the first two parts of the discussion. Now we go ahead with the vaccine development strategies. So, what is the vaccine? Vaccine, as we all know, is going to, in simple words, if I want to say, it is going to trigger our immune system by introducing an antigen, or say it as a spike protein present on the surface of the virus, SARS-CoV-2 virus in this case, and that is going to act as an antigen here, so that we are actually making our immune system realize that this kind of protein, you may interfere in future, so be ready for the attack. Okay, so that is what, in simple words, the vaccination is a part of active immunity. So since so many years we have been developing vaccines, but the development of vaccine depend upon what kind of strategy we are using. So the strategies are clear cut, divided into two categories. One is classical, wherein we have created attenuated or weakened viruses, and a very good example of this is MMR vaccine, which stands for measles, mumps, rubella vaccine. Then we have also created inactivated or heat killed viruses, which are given as a vaccine. A good example would be polio salk vaccine and hepatitis A or rabies vaccine. There are vaccines created with the help of virus-like particles. The Gardasil is the product given for human papilloma virus, and protein subunit vaccine in case of hepatitis B virus is also being administered, which is also coming now under classical. But now we have something called as next generation of vaccines or the strategies. So the first and foremost is viral vector, wherein we use a replicating or a non-replicating viral vector for administrating genomic content. Then we also have nucleic acid based vaccines developed, which could be directly incorporating DNA or RNA into the host cells. Then we also have protein based vaccines, and more with respect to COVID-19, we have more amount of protein based vaccines getting created. And we also have antigen presenting cells used as vaccines. So, um, in general, if we talk about the vaccine development, the main true 2 points that are important for vaccine development are: one, that we do not want any side effects of the vaccine, and second, we want it to be highly effective. Okay, now effectiveness of the vaccine and efficacy of the vaccines are the two terms which are generally used interchangeably, but there is a very slight difference between the two words, which we would talk about little later. Apart from these two parameters, we also want our vaccine to be very cheap, which should be easily administered. Nobody likes to take an injection. So generally, vaccines which could be oral or which could be intranasal, that are preferred over the ones which need to be given intramuscularly. Also, we don't want to take, if it is an injection method, we also don't want to take the booster dosages again and again. So number of dosages as less as possible would be preferred. So these are the countries, which is a map of overall forerunners of the race of the vaccine development. And you can see the North America, India, China, UK, Germany, and Australia. These are few countries which are in the race for development of vaccines, and you can also see their strategies given around. We will take a broad look at all these strategies, but this was just an overview. And as considering the strategies used, you can see that conventional strategies are far lagging behind than the ones I talked about under next generation vaccine development. And that too, protein based vaccine developments would be at a higher rate, especially because spike protein is the one is being targeted here in this case. Okay, with this, we will now learn about what are the stages that the vaccine development process has. The first and foremost step is the preclinical step, in which we study all the effects of vaccine development and administration of vaccine on animals. Okay, so mice is the preferred model animal, and then it goes. I mean, once we are assured that everything is working well in mice, then we start with human trials. So there comes clinical trial phase 1, where we test it on the human lesser than 100. So number of people that are getting vaccinated in this trial are less than 100. The LL and UL represents the lower limit and upper limit. So how much of the vaccine to be administered, so that not too low, not to have its effect, but not too high to create adverse side effects. Okay, so this has to be studied in phase 1. Then we look for side effects, what kind of side effects we get, and we need to standardize the doses. Then we enter into phase 2 clinical trials. Phase 2 clinical trials, we are increase in the population, the 100 to 1000s, thousands of, it could be 5000, it could be 7,000, 8,000, but in thousands. And apart from SE, that is side effects and doses, we also check for the demographics of the population. That is, with respect to age, we have to differ. We have to give vaccine to all age groups, and maybe the race could be another criteria, the geographic location could be another criteria. And that is why if you read different reviews on COVID-19 vaccines, you would understand that trials are begun in different countries. Even if the manufacturer is situated in one country, they are not restricting their trials in only their country, isn't it? So and the third phase of human trials will consider 10,000s of population. It could be 10,000, 20,000, 30,000, maximum of 60,000 I have come across in the reports. So again, the same demographics, again we have to recheck, and side effects, again we have to take a monitor these side effects, and we have to also talk about the actual doses that needs to be given. Apart from this, there is something peculiarly done in this phase. That is, we talk about real life situations. Like these people who get vaccinated, they they are allowed to do their routine activities. Okay, um, they are not kept under clinical controlled atmosphere. They, because ultimately vaccine is going to be given to the common people, and they are going to do their regular activities. So we have to check in this phase whether everything is working fine or not. Okay, and then once it is all fine, then we go in for approval. Generally FDA, Food Drugs Administration, is the um, company that gives approval. But under the situations like pandemic, we can have approval given by in the under EUA, that is called as emergency use authorization. So once understanding these stages, we now understand that these stages are very, very critical, and they take lot and lot of amount of time actually to proceed. So the preclinical and every phase, if you see, hardly it is taking 2 to 3 years of um, um, stringent process. And so, as a ideal condition would be 10 to 15 years for one particular vaccine development. Then you would be surprised how come the COVID-19 vaccine is being available within a year of span. So here is the answer: because we, the under the rapid requirement of vaccine, companies have not waited for first phase to get over and then start with the second phase. You can see almost in every phase it has started in the middle of the earlier phase. And because of this combined phase trials, it is the rapid processing of the authorization that has started. And you can see even manufacturing starting with the start of phase 3 trials. Okay, now this particular diagram will give you an idea or an overview, like what company, what manufacturer is utilizing what strategy for vaccine development. Most of the strategies we will talk about um, one by one. So the first vaccine, which is the Moderna vaccine, a US-based biotechnological firm, and its vaccine is called as mRNA 1273 vaccine. And these are the collaborating institutes with which it has created this vaccine. Now, what is the strategy that this vaccine is using? That it has got a delivery system. This delivery system is a nanoparticle and a lipid nanoparticle, LNP, short formed as, and inside that we have put up mRNAs of the spike protein that we discussed. And when it goes inside it, it is delivered inside the host cell. The host cell will take up that mRNA and will get translated. The mRNA will get translated, and finally, instead of virus, only the spike protein will be represented on the surface. Now the immune system will come into picture, and it will recognize, oh, this protein is something new. So it will recognize and it will create antibodies against it. Not just a B cell, there is also some T cell response associated with the protein exhibited on the cell surface. But we will not talk about the immune response elicited in this particular section, because we will confine ourselves to the vaccines against COVID-19. Okay, so this is the strategy, and this is how the lipid nanoparticles look like. They themselves have a lot of advantages for their use into drug delivery. um, I will not go into the nanoparticle section because that's also a huge field to talk about. Considering our topic, I will go ahead with the other vaccines. On the same line, the BioNTech and Pfizer, they both have come up with another vaccine called Tozinameran, and the brand name is Comirnaty. And the only difference between the two vaccines are the storage conditions, as you can see, and also the difference between the how many days between the two doses. Otherwise, the strategy is one and the same. This was the first vaccine to get approved. On 2nd December 2020, it got approval for the first time. Now let us understand the third type of vaccine, which is developed by AstraZeneca and Oxford University, and CHADOX1 is called, why it is so, because CH stands for chimpanzee, AAD stands for adenovirus, OX stands for Oxford University, and that vaccine is actually called as AZD1222. Its brand name is Covishield. Now, what is the strategy being used in this vaccine? As you can guess, that we are using chimpanzee adenovirus. Now, to treat viral infection, use of another virus. How come? Just look at the diagram. The virus that we are going to use, chimpanzee adenovirus, is going to get modified. We are modifying this virus in a way that it is not able to cause the disease. So this becomes our vector. Okay, we are going to use it as a vector as in the carrier molecule, just for the delivery of whatever material we give inside its structure. Okay, so earlier two vaccines had lipid nanoparticle as the carrier. Here we are using another non-replicating adenovirus as a viral vector. On the other hand, this is the SARS-CoV-2 virus. From this virus, we are going to take out its genome, say reverse transcribe it to get cDNA. Once we get cDNA, we are going to convert it into double-stranded DNA. so that we get the coding sequence of the spike protein, and that sequence we are going to put it into this chimpanzee adenovirus, modified chimpanzee adenovirus, and this we are going to inject. Once inside the host cell, given by intramuscular injection, the delivery will take place, and the spike protein will eventually be generated, and it will create immune response inside the body. So this vaccine also got an approval 30th December 2020. And a very peculiar thing about this vaccine was that there are two things. One is, it can be stored at 2 to 8 degrees Celsius. That is a normal refrigeration condition. Now, why this? Because we are not using lipid nanoparticles. Lipid nanoparticles are uh, very delicate kind of particles. So we have to preserve that. For their stability, we have to preserve those vaccines at minus 20, minus 80 degrees Celsius. But this is not the case here. We can use normal refrigerator for the preservation. Very interesting story behind the doses and efficacy of this particular vaccine, that generally all COVID vaccines have two doses to be given at maybe 28 or 21 or 14 days with a gap. Okay. So, what happened when the clinical trials were carried out with this vaccine? By mistake, okay, by mistake, the first dose was given only half of the required quantity. Okay, so suppose 1 ml of the formulation has to be injected, so zero point 0.5 ml was injected by mistake. Okay, it came as a surprise to all the scientists that when someone injected a proper amount for the first dose as well as for the second dose, the efficacy of the vaccine was 62% only. But with the mistake, the efficacy increased up to 92%. And that came like a positive thing. Although AstraZeneca is not able to yet, I did not find any reports noting down why and how it must have happened. Uh, they are still finding out it, but that's a good news, isn't it? On the same line, Moscow's Gamaleya Research from Russia has come up with another vaccine with the same adenovirus, which is called as Sputnik 5. Only change in this vaccine is the two doses are given with the two separate adenoviral vectors. The first dose given with adenovirus type 26, and second dose is given with adenovirus type 5. Okay. Otherwise, rest of the procedure remains the same. Now let us talk about some conventional strategy being used for vaccine development. I hope you remember conventional strategies or the classical strategies involved heat killed or life attenuated viruses. So this is how we have the healthy virus getting converted into inactive virus, which is not able to cause infection. But as you can notice that its spikes and everything remains same or looks same, and we don't want to denature its spikes because those are the ones going to create the immune response. So there are different ways to have inactivation of virus done. We can use heat, low pH, alkali, detergent. Many treatments can be done. And using this strategy, India has come up with a vaccine called Covaxin with a Bharat Biotech situated in Hyderabad. They are also planning to set up their second plant in Odisha, and this vaccine got a very recent approval on 2nd January to 2021. Uh, on the same line, Sinopharm vaccine from China is also going to be soon in the market, which also utilizes the inactivation of virus strategy. So this is an overall chart or an overview for the front runners of the manufacturers for COVID-19 vaccines and their comparison. These are some of the candidates which are still in phase 3, 2, or 2, 3. Like I told you, combined trials have been undertaken. So These are the candidates which you will have soon uh, available in the market. Covivac from Serum Institute India, which is actually a live attenuated vaccine and a new strategy. And one good thing about this vaccine is it is going to be only one dose required, and it is administered intranasally, while all other vaccines were administered intramuscularly by injection. So, this will be, I think, a preferred route of administration. Similarly, CanSino Biologics from China is also coming up with a vaccine, and USA is trying, Novavax is trying the recombinant protein strategy for creating vaccine. There are two DNA vaccines in line as well. So I was talking about efficacy of the vaccine, isn't it? So whenever in a trial we consider, suppose I let us take an example that it is clinical trial 2, and I told you that we have some thousands of people, isn't it? So 100 to 1000. So suppose I have 1000 people to vaccinate in for the clinical trial 2. So what I do is I divide the population equal halves. So 500 people on the left side, 500 people on the right side, and 500 on the left side I give them placebo. Now placebo is not actually the vaccine, uh, it is a saline. Okay, so we are not giving these people vaccine, but they won't be told. It's the studies are generally blindfolded, so they are not told. They are psychologically told. They feel that they are vaccinated. Okay, but these people are also a part of study. We do monitor their changes, and other 500 people would definitely be vaccinated. And at the end, at the end of both the doses, we calculate how many positive as in how many people developed side effects or severe illness or any kind of abnormality that you would find. So suppose the placebo given a vaccinated people, should I say, or unvaccinated people, they got 20 people out of 500, they gave us positive reaction. Vaccinated people gave us 4 as a positive cases. Then how do we calculate efficacy? We negate the positives of vaccinated from unvaccinated and calculate its percentage with this formula. So here in this case, in a hypothetical case, we have got an efficiency of the vaccine as 80%, right? So now you have full understanding of how to calculate vaccine efficacy. But this is all calculated under clinical trials. The effectiveness clearly means that um, the vaccine, how it well works without the clinical trials. So when it is given to the population and not being monitored clinically. Okay, so that actually tells you what is the effectiveness of the vaccine. So, with this all discussion about different vaccines, we still have certain challenges in vaccination. There is a lot of news, a lot of articles, and media, and everything is, information has become so uh, well exchanged nowadays that people listen to something, hear something, or read something somewhere, and it creates a panic very easily. It is transferred also very easily nowadays. So there is a lot of problems with the communication and trust because people are saying that how come you got a vaccine within a year, and that's a good, I mean, a pretty valid question when it used to take 10, 15 years earlier. Now, how can you get it within a year? So distrust is what people are showing over vaccines, and yes, there is a problem with funding and distribution of the vaccines that have been created all over the country. There are certain side effects seen which are mild, but yet they are seen, and there are reports. Um, there are disparities with racial and ethnic concerns, and logistic and supply of vaccine is going to remain a problem in future. Uh, Certain manufacturers are also stating about some political pressures on them, and of course the coverage and costs of the vaccine. So these challenges remain still a part of vaccination drive, but let us hope to get rid of this SARS-CoV-2 very soon. With this, I end my talk. These are some of the references I referred to, and thank you very much. If any questions, doubts, comments, most welcome in the comment section.